When we think of purity, we often think of water, especially mountain water. But on its journey to the ocean, mountain water undergoes many changes. It is transformed from something to enjoy, touch, experience and admire, something that is brought into our valleys unspoiled, into a commodity to meet our needs. Into highways of wastewater transporting our pollution away from us. Pharmaceuticals, for example, micro-pollutants that, even in very low concentrations, can harm the environment. Our society uses more and more pharmaceuticals, chemicals designed to have an effect on our body. And these pharmaceuticals are increasingly polluting our environment. When we take medicine, only a small part actually remains in our body. Most of it, in some cases 90%, is excreted. It ends up in wastewater and is transported to the sewage treatment plants. But most sewage treatment plants do not eliminate pharmaceuticals. This is why they find their way into our surface water. And because pharmaceuticals are intended to interact with organisms, they may affect the organisms living in these waters. It has been shown, for example, that estrogen can feminize male fish, causing infertility and even the collapse of fish populations. At the Institute of Environmental Studies at the Free University in Amsterdam, scientists are looking at the effects of different pharmaceuticals on fish.
I'm working on a EU project called Farmers, and within Farmers we study antibiotics and anti-cancer drugs. And we do that because we think these compounds are not easily degraded in the environment, and they are increasingly used in hospitals, and in case of the antibiotics, also increasingly used in food production sites. We think that these compounds might not be catched with normal sewage treatment plants. And as an example, for some anti-cancer drugs, we know that they are used up to 5 kilograms a year in one single hospital. And so far, we don't have any environmental legislation which controls or monitors these pharmaceuticals in the environment.
Zebrafish are a valuable model system for scientific investigation. This is because their physiology, metabolism, and genetic makeup can help us understand what is happening inside other organisms, including humans. Another trait that also makes zebrafish a good subject to investigate is that their eggs are transparent and consequently suitable for developmental observations. Therefore, the researchers in Amsterdam start their work with the collection of fertilized eggs from the fish they breed in their aquariums. After sorting, the fertilized eggs are exposed to various pharmaceuticals in different concentrations. The aim is to find out at which concentrations a certain chemical is harmful to the organism. Step by step, the scientists can raise the concentrations of pharmaceuticals until a harmful effect is observed.
With our work, we hope to assess the risk of the pharmaceuticals on the living organisms and the environment and humans, and we hope that our results will also give advice to decision makers.
To assess the risks pharmaceuticals can pose to living organisms, scientists can examine the fish under the microscope. Here, they can check the proper development of the cardiovascular system, the nervous system, and the functioning of their organs, such as the eye. When the embryos hatch, their behavior, for example their mobility, can also be observed. If it is abnormal, it may indicate genetic defects. In that case, scientists need to find out which molecular changes are responsible for the observed behavior. But fish are not the only organism that should be investigated.
In order to get a real picture what the pharmaceuticals are doing in the environment, you have to look at all different organisms in your water environment, starting from the bacteria, to the algae, to the water fleas, and to the fish.
Pharmaceuticals are only the tip of the chemical iceberg that can affect living organisms in our waters. Thousands of chemicals are discovered every day, and thousands of chemicals are used every day. Detergents and personal care products in our homes, lubricants in industries, and pesticides in agriculture. All these different chemicals can find their way into our surface waters. Here, alone or in combination, they can cause harm to the aquatic ecosystem communities, even in low concentrations, even at less than 1/1000th of a gram per litre. In the Gullmars fjord in Sweden, scientists are studying the effect of pharmaceuticals and other micro-pollutants on biofilm communities. As the fjord is still almost free of pollutants, it is an ideal environment for the scientists to cultivate unpolluted biofilms for their research. But they have to go out regularly to check and collect them. Biofilms are communities of different living organisms such as bacteria, algae, and small invertebrates. They fulfill crucial ecological functions in the water. They carry out photosynthesis, degrade compounds, or serve as food for other organisms. Here, they are cultivated on glass slides and suspended in the water to be later used in laboratory experiments.
The experimental strategy that we are following is a combination of fieldwork out in the fjord and exposure under controlled laboratory conditions. We are interested in the effects of toxicants like triclosan, the personal care product that you find in toothpaste or soap, for example, or antibiotics, but also heavy metals or something like that. And we want to see the effects of those compounds on natural communities that you're finding outside. So we are giving those creatures the possibility to colonize artificial substrate, those small glass discs. They colonize that, and then if the community, or as soon as the community has established itself, we take it into the lab, where we then can expose that under controlled conditions to a concentration series of those chemicals in order to see which concentrations are harmful, which concentrations are not.
Scientists don't only study the effect of individual pharmaceuticals or other pollutants on biofilm communities, they also investigate the impact of different mixtures of pollutants. The experiments are carried out in small aquariums that closely reproduce the conditions found in the environment. The biofilms on the glass slides are immersed in the fjord water and exposed to different combinations of pollutants.
We have to consider that pharmaceuticals are not acting on an environmental organism singly as an isolated individual chemical. What we usually have is a whole cocktail of different compounds, and not only pharmaceuticals, but also biocides or pesticides or industrial chemicals, and all those chemicals are acting together on the exposed organisms. And when it comes to mixture effects, what we see is that the toxicity of the mixture is driven by the potency or by the toxicity of the individual compounds, by their concentration in the mixture, and by how many compounds are in the mixture.
After exposure to the different compounds, the Swedish scientists evaluated the changes in the different layers of the biofilm community. They observed that some organisms in the community were sensitive to the chemicals and had died. Others were tolerant and had survived, changing the community composition. These effects differed depending on which compounds, for example pharmaceuticals, were used in the experiments.
When you're asking about the groups of pharmaceuticals that we should be worried about, I think We have to actually take a close look at those pharmaceuticals that we are developing for killing certain organisms. For example, antibiotics are meant for killing bacteria, so we actually need to check what's happening with environmental bacteria.
The release of pharmaceuticals into the aquatic environment can have an influence on which organisms will survive there and which will die. So, when, for example, bacteria come into contact with antibiotic drugs, only those bacteria resistant to one or more of these drugs will survive. The antibiotic-resistant bacteria will populate and dominate such an environment. If they become resistant to a number of drugs, they may even pose a serious risk to our health. Antibiotic-resistant bacteria have already found their way into the wastewater streams of our cities. Can they also find their way from the cities into the fresh waters of rivers and lakes? What could happen if they do? Scientists from Switzerland and Germany wanted to find out. Nadine Cikalski was one of the principal investigators in a study that was carried out on the shores of one of the largest freshwater reservoirs in Europe.
I investigated the sources and the spreading of antibiotic-resistant genes and bacteria into Lake Geneva, Switzerland, especially the Weedy Bay, and I was interested in what will happen to them as soon as they arrive there.
Nadine and her colleagues took a close look at locations where bacteria may become antibiotic resistant. The hospital, the city canalization system, the municipal wastewater treatment plant, and they took a look at where antibiotic-resistant bacteria may end up, the lake.
Due to the immense use and also misuse of antibiotics in human and veterinary medicine, there is increasing problems with antibiotic resistance, especially with respect to pathogenic bacteria. However, what is a quite new aspect is that you find resistant bacteria and resistant genes in wastewater, and then the question more and more turned up. So these wastewaters are discharged into the environment. What happens in the wastewater treatment plants? And if nothing really happens, so what will then happen if all this ends up in the environment?
To find out, the scientists first collected samples. Wastewater samples from the city, as well as water and sediment samples from the lake. Collecting samples from the lake was the biggest challenge, especially when it came to operating a special corer to take sediment samples from up to 50 meters in depth. First, the scientists had to lower this heavy instrument to the bottom of the lake, so it would, with the force of gravity, penetrate the sediment. Then they had to make sure that the corer brings the right amount of sediment back to the surface, so that they can conduct further analysis in the lab. Will they find antibiotic-resistant bacteria in their samples from the lake and the city? And will multi-drug resistant bacteria, particularly dangerous for humans, be amongst them? To answer these questions, bacteria were extracted from the samples collected at each of the test locations. Under sterilized conditions, these bacteria were plated onto different Petri dishes containing different antibiotics. The Petri dishes were then incubated at a temperature of 37 degrees Celsius. After a few days, the cultured bacteria were ready to be evaluated. And it could clearly be seen that not all of the bacteria had died when exposed to antibiotics. Some had survived. Therefore, some of the bacteria extracted from the samples in the city and the lake were antibiotic resistant. But not all bacteria resistant to antibiotics can grow on a Petri dish. So a second test was necessary to identify them. This test allowed the identification of DNA fragments and sequences of antibiotic-resistant bacteria. The results of the Petri dish and DNA analysis combined showed that antibiotic-resistant bacteria, including multi-drug-resistant bacteria, were found in all of the samples that had been collected, from the city and from the lake. What surprised the scientists was that after passing through the wastewater treatment plant, the proportion of multi-drug resistant bacteria in the treated water had actually increased. How could this have happened? The close proximity of bacteria to each other at the wastewater treatment plants could have been a factor. One or more antibiotic-resistant genes could have been transferred from one bacterium to another by 3 different routes. First, by conjugation. Here, the bacteria are in physical contact with each other. The DNA containing the antibiotic-resistant gene is copied into a bacterium that does not have this gene. Second, by transformation. Here, a free-floating DNA containing the resistance gene is taken up by chance by a bacterium that does not contain the gene. A third way in which resistance genes are transferred is by transduction. Here, viruses take up the resistance gene from the bacterial DNA and transfer it to other bacteria. Micro-pollutants have been found in the wastewater of our cities, and if they're not removed, they can also find their way into the fresh water of our rivers and lakes. It is one thing to consider the impact of micropollutants such as pharmaceuticals on living organisms, but quite another to consider how these affected organisms behave in nature. Both the micropollutants and the affected organisms may turn out to be a serious problem for us in the future.
For me, it is clear that I guess the main steps that will come in the future will that wastewater treatment plants and probably also drinking water treatment plants will get an additional treatment step in order to remove micropollutants such as antibiotics.
I'm living in this environment, my family is living in that environment, and maybe later on my kids will live in this environment, and I think they should have a safe environment to develop. So that's why I think it's very important to really study impacts of pharmaceuticals or other pollutants on the environment.